Good morning, students. Today we are going to see about the nanobiotechnology topic. So, about the introduction about nanobiotechnology. Nanobiotechnology it's a combination of two progressive fields. So that is nanotechnology is one field and the biotechnology is another field. That combinations they are using the name of nanobiotechnology. So nanobiotechnology is a division of nanotechnology which uses nanoscale of the material and then biological is a starting material for the nanobiotechnology. So it uses tools of nano microfabrication to build device for studying biological systems. Then it keeps existing opportunity to bring high impact advance in the field of bioengineering and then biomedicine. This is the main focusing for nanobiotechnology here. So nanobiotechnology mainly provide the structural features of biological systems such as cell, tissue as well as to develop nanobiomaterial for the diagnosis, therapeutic and tissue regeneration. In the application of nanobiotechnology in the field of biology and medical science is very high. So ongoing work including the development of nanopores, it be characterize the nanoscale structure and then the function of the cells. So here this slide we have to represent what is nanobiotechnology. Nanobiotechnology we have to define and the creation of fundamental materials. We have to create that material or device or system through the understanding and control of matter at nanoscale. That synthesis the material device system comes under the range of 1 to 100 nanometer is there, which a new functionality and properties of matter absorb and host for a broad range of applications. So here we have to represent the some of the nano creation material used for the many fields. That's a engineering field we have to use and then tiny motor system and then semiconducting metal junction formed two carbon nanotubes. And then intro to the nano. Nano means the meaning of the dwarf. So it comes under the 10 to the power of minus 9. That is a meaning of 1 billion. So nanometer million part of the meters. So idea was presented by Richard Feynman. The average page is about the 1 lakh nanometer thing. A very fine human hair is about the 10,000 nanometers wide. So the scale we have to represent here for the nanometers comes on the range of the 10 to the power of minus 9. So here everything we have to present 10 to plus ranges. Here after 1 to 10 to the power of minus 1 which comes under the family of minus ranges. So anything below 100 nanometer is part of the nanoscales. So water comes under the 10 to the power of minus 1, glucose is plus 1, antibody 10 to the power of plus 1, then virus 2, bacteria 3, cancer cells are 10 to the power 4 to 5, and then any period 10 to the power 6 and 7, and tennis ball comes under the 10 to the power of 8. So below less than of the 1 to 100 nanometer we are called as nanoscales. So here we have to represent the definition of the nano technology is the understanding and control of matter at dimensions of roughly 1 to 100 nanometer where unique phenomena enable novel applications. So encompassing the nanoscale science, engineering and technology, nanotechnology involves the imaging and measuring, modeling and manipulating matter at this length scale. So why nano is very important? Because that is a small, that is enough to enter into any of the cells. So we have to use that's why nano material is a main important field. In the nanoscale devices can easily enter the cells and organ to interact with the DNA and protein. May also able to monitor the entire entry of the cells into the living body systems. So here the cell, this is a nano device. How it will be enter into the cell? We have to directly monitor the nanoscale level. In the nanomaterial synthesis, there are two way we have to synthesis. One is the bulk nanoparticle, another one is a surface. So bulk is a one phase or a multi-phase method are there. Surface structural surface or film or structural films is there. So the particles, every newly synthesized nanoparticles structure and then properties will be changed based on the parameters. Here we have to use two methods, top down and then bottom approach for the nanoparticle synthesis. The bottom up including the natural self-assembly and technological self-assembly, UV lithography. Top down methods which comes under the electron beam lithography, nano imprint lithography, scanning probe in lithography. So here we have to represent some of the properties of nanomaterial. If suppose we have to synthesize any nanoparticle, that nanomaterial have some of the property. The properties mainly, these are the main things. One is a catalytic property, another is a electrical property, another one is a magnetic, and then important properties a mechanical, optical, spherical, and then biological. So these are the main properties we have to synthesize any nanoparticle in the state. So synthesis nanomaterials having the many shapes. One is a nanorod, another one is a nanotube, another one is a nanocomposites, another one is a nanoparticle. These are the main nanomaterial shapes available in the system.